Hello, everyone. Thank you for joining us here today. I'm Sue Romanoff, Head of Healthcare Content at Edison Group. Today, we're kicking off the Oncology series with this webinar. Oncology remains a very important area of discovery, and the first of our series covers oncology vaccines, which the title suggests is highly specialized, but likely a timely topic nonetheless. You can also refer to our note if you are interested in more details at the Edison website. Today, I have the pleasure of introducing two highly experienced oncology experts focused on oncology vaccines. Jens, do you mind giving us a brief introduction?
My name is Jens Bjørheim. I work in a company, Ultimovacs, which is located in Oslo, Norway. We work with cancer vaccines, and our lead compound is a UV1 vaccine based on telomerase sequences. So it's a peptide vaccine with long peptides, and we are currently in phase 2, 5 different indications, randomized trials, one in malignant melanoma, one in mesothelioma, ovarian cancer, non-small cell lung cancer, and head and neck cancer. The company as such is not that large, 25 people working with these clinical trials. We expect a readout of the first 2 trials early next year in malignant melanoma and mesothelioma. Up until now, we have conducted 4 phase 1 trials in different indications to identify the right dose for the UV1 vaccine, and also in different combinations. Moving forward, the most exciting thing for us will be the first part of next year, where we have the readout of the 2 first randomized trials.
Jeroen.
My name is Jeroen Rovers. I'm the Chief Medical Officer for Mendus. Mendus is maybe not a familiar name because it was basically introduced just before summer, but it's the come together of 2 companies. One was DC Prime from the Netherlands, privately owned company, and the other one was Immunicum, a Stockholm listed company. I think what brought these 2 companies together is what we're also probably discussing today. So what we are developing is immune primers based on dendritic cell biology. And our lead product is DCP-01, which is a cancer cell-based immune primer, which we use to trigger a systemic immune response towards several types of cancers, and specifically hematological malignancies. And that's most advanced in the development for a AML maintenance treatment. But additionally, we also have a donor-derived immune primer, which we use to trigger an immune response within a tumor, leading to the attraction and activation of immune cells, such as NK cells. So that's what we are doing, and we're in phase 2 for both projects, and hopefully soon we'll be able to report a bit more on our AML program.
Great. So this is, it's such a big topic, so I think we can start really basic, if that's okay with both of you. The field of immuno-oncology treatment has advanced significantly in the recent years, particularly in cancer vaccines. While success has been limited for earlier vaccines, we are now seeing some exciting clinical results from the new generation of cancer vaccines. In your view, how has the understanding evolved to bring us to this point of clinical breakthrough? Jeroen, maybe you could start there.
I think one of the things which we know and what we've seen is that a lot of these approaches, if you call it, say, vaccine approaches, were used in oncology, but often in advanced stages of disease. And I think one of the downsides of an advanced stage of disease is that either, say, the tumor suppressive environment is to such a degree that any response from the immune system might be difficult to generate and trigger. And secondly, there are always in patients which you have to question whether, say, the immune system might not be, say, exhausted already, and is also from that perspective not able to react. So I think what we currently are seeing is that if you move the timing of your, say, approach. So when you want to, say, trigger the immune system, you need to do it at an earlier stage in the disease. Ideally, of course, a vaccine is to prevent something to happen in the future. But for cancer, that's very difficult. I mean, we don't know which type of cancer we will get or which subtype or so. So you cannot vaccinate to prevent the cancer, but you can vaccinate to trigger the immune system to respond to the cancer you have at that moment in time. And what we are doing is to just use this principle to start, say, treating and prime the immune system as early as we can, meaning that the bulk of the disease is under control and that you have a chance at that moment in time to give the immune system the reaction you need to have a durable response.
Great. How about Jens?
So I totally agree with Jeroen's considerations around this. I would like to add the checkpoint inhibitors, at least for seeing the clinical efficacy. So I guess we all can agree that a pre-existing T-cell repertoire is needed in the body for the CPIs to show clinical efficacy for patients. So in some patients, that repertoire will be sufficient for the patient to get into a complete response or a partial response. But for most patients, the T-cell repertoire is not large or selective enough to kill off the cancer. So in our trials and in our view, it's important also to combine with checkpoint inhibitors. We believe that there is a mutual dependency between the immune system on one side and the checkpoint inhibitors on the other side that hopefully will lead to relevant efficacy for patients in different indications. I also do agree that it's also time to look early in the early stages of cancer. In our company, since we are combining with the checkpoint inhibitors in all indications, we have been mostly in the advanced or metastatic setting. But of course, now with the new interesting results being presented also in the early stages and the neoadjuvant setting especially, and the early settings are very interesting for a therapeutic cancer vaccine.
Much of your work tests the conviction of your theory over a long and arduous path, trial by error, really to prove your assumptions. So maybe it makes sense to take a minute to understand your precepts and key objectives for your oncology vaccines. There are many angles, and we go through some different categories in our note, but perhaps I can interject a few thoughts. Does your vaccine have targeted or broad approach or specific indications or personalizations, or do you look for something else? Jens, could you start on that one?
So our company work with self-antigen telomerase, which is a naturally occurring protein in the body. It's not expressed to a large extent in wild-type tissues in grown-ups. What we find interesting with telomerase is that it's a prerequisite for the cancer. So 85 to 90% of cancers, they have started the expression of this protein or enzyme to be in a position for eternal cell division. So it's something that is needed to be there up front or early in the cancer development, and it will be there throughout the course of the cancer and in all parts of the cancer. And the cancer cells cannot stop expressing telomerase. So it's a relevant antigen throughout the course of that cancer. And it's nothing with the telomerase itself that we are targeting, just to be precise on that. But we know that the tumor cells will continue to express telomerase over time. And therefore, the T cells we expand will recognize their target in the tumor and in the draining lymph nodes in all stages of cancer.
Jeroen, do you want to jump in here now?
Yeah, I think, like you said, our approaches are distinctly different. And so I think what we are doing is take a different approach. So we use dendritic cell biology. So we know that dendritic cells are key to also orchestrating and also, say, managing, say, immune responses. But we use that specifically to make our product immunogenic. So the basis of our product is a cancer cell. It's once derived from a patient suffering from leukemia. And we've got the technology to make that cell differentiate into a dendritic cell. So it's got all the phenotypic properties of a dendritic cell. And what that does is twofold, but the main reason is that it triggers any, say, immune response if you put it in contact with the patient. And it's an allogenic approach. So we have an allogenic off-the-shelf approach. So the allogenic element there is also relevant to trigger the immune response. And that makes it distinctly different. So what we have is antigens, which are leukemia derived in this case, but they're more applicable to a broader, say, variety of tumor types. We're looking at, say, WT1, RAM, PRAME. They're all contained within this leukemic cell, but we offer them in the context of an allogenic cell. So often, of course, you have to question yourself, we know these tumor-associated antigens are there. Why is the immune system not responding? Well, probably because in an auto setting, there are these mechanisms trying to prevent reactions towards something you don't want. But here, this is totally off. So what we provide is antigens in the context of an allogenic approach with dendritic cell phenotype, triggering basically all the alarms you can get in an immune system. And that translates into the response. And it's distinctly different from what some people call, say, dendritic cell vaccines, because we don't use the dendritic cell to be, say, the functional element here. We just use this cell to be the immunogenic, say, trigger to react towards the leukemic antigens. And that makes it distinctly different.
Yeah. I think it's important to emphasize the key objectives of vaccines, maybe just to start off. So I think vaccines prime the natural immune system, and they also create memories, and they help monitor to create a faster response in the future. Maybe does this get around that resistance from the other approaches? And Jeroen, maybe you could kind of start on that one.
The resistant part is to be seen. I think that's a difficult one because it all depends where you trigger your reaction towards. So we're working in our lead product in acute myeloid leukemia, and that's a very heterogeneous disease. So if you would want to trigger, say, a response towards a specific mutation, then you always have the chance that that clone might be successfully treated, but you can get another clone of AML to emerge again. And that's the reason why we don't have a, say, targeted or tailored kind of approach. The cell we use, we know, contains a large variety of tumor-associated antigens, known but also probably unknown. So there will most likely be new antigens into that in that cell. And it triggers a very broad immune response, which hopefully then controls all the different, say, subclones you might have of such a disease. In a disease where it's more clear cut, where you have, say, one target, or where you can just get control over that and then be done for, say, the rest of your life, it's a different approach on how you look at that, I would say.
Jens, you want to chime in on that one?
It's a tricky area, as Jeroen said, so it's a lot of things to discuss here, but I will try to make it simple and from the place we are in development. So as I said, we are combining with checkpoint inhibitors. So if we look at the curves in a typical indication where checkpoint have efficacy, you will see in a way three large groups. One group of patients that doesn't respond to the checkpoint inhibitor at all. They will progress quite fast, so-called primary resistance. We do not believe that vaccines as such will do the trick here for most patients. There might be individual patients that you can turn around to get an efficacy, but for the most, these with primary resistance, they will maybe not be helped by new T cells coming along that also could be produced by the body itself. Then the best group, those with a complete response, you can assume that those patients had a resistance that were unblocked by the checkpoint inhibitor, and that the T cell repertoire in those patients were sufficient to kill off the cancer. But then you have this middle group of patients, those that go into some stable disease or partial response and progress after a while. There you, I guess, can find several different explanations for that happening, both the development of secondary resistance, HLA molecules might be affected, and other mechanisms. But on the other side, also that the T cell repertoire in those patients are not sufficient to keep the patient into a response, and also not to push them further into a complete response. So, and in this sense, at least we in Ultimovacs, we think it is important with a target that is present in all parts of the tumor, both in the primary and in the metastases. We know that the tumors are heterogeneous, but we also know that telomerase is part of the trunk, in a way, of the cancer, represented in all parts of the tumor. So if the immune system get interested in the tumor, we expect efficacy in all parts of the tumor, both the metastases and the primary.
One element to add there, I mean, we're talking about triggering an immune response and then or activating or releasing the T cells to destroy the cancer cells. But one element which we know is also still important, you need to be able to get them into the tumor itself. So we know there's some tumors which are very difficult to allow the T cells to get in there. So I think that's a key element. And one of the approaches we're trying to develop as well is that we have an intratumoral approach where we inject these primers directly in the tumor, and that's to hopefully then attract more immune cells to get into the tumor to do their job. I think that's also a relevant element here.
One extra comment regarding this resistance. So I will try not to make it too complicated, but the UV1 vaccine today, we call that a second generation vaccine, obviously because there is also a first generation telomerase vaccine that was called the GV1001, and it was investigated prior to the CPIs on the market, so as monotherapy in different indications. Around 1,000 patients in different indications were treated with this peptide, a single long peptide from telomerase, and without CPI. What we saw, as maybe we expected, we saw good immune responses in patients, but little efficacy for patients. But in some few patients, we did see very long overall survival. And when we analyzed the T cells from these patients afterwards, we saw that they had expanded T cells to different parts of the telomerase. What I'm trying to say here is that in some patients, this antigen spread occurred, that the body itself were put in a position to expand T cells that were relevant for that patient. And the UV1 vaccine as such is actually based on 3 such fragments that were identified in those with a long-term survival. We did not see such expanded T cells in those with a shorter life survival.
So both of you are kind of, you had ESP there because I was getting to immune checkpoint inhibitors next here. So I think they've had a really profound impact on the shape of oncology treatment, that in general, the whole landscape, right? But I don't know that they've lived up to their hype. Logically, it seems like vaccines would naturally complement checkpoint inhibitors by priming the immune system, while ICIs prevent turning off the tumor immune system resistance. Is it naive to assume that the combination therapy would pave the way for future clinical success? Jens, maybe you could start there.
Well, so we think the relationship or combination treatment with checkpoint inhibitors are very important. And it might be so that there are different checkpoints not identified as of today, where we don't have drugs. So it could be parts of subpopulations or in indications we do not have the proper immune therapy as of today. But I think that the large phase 3 trials with checkpoint inhibitors that show effect on patients, they are in a way guiding us, because the only thing that leads to an effect for those patients are the immune system that are pre-existing prior to the checkpoint treatment. So in a way, following the checkpoint inhibitors is, for us at least, very valuable in understanding where to go. Unfortunately, we cannot go, for example, into prostate cancer as of today. No checkpoint inhibitors are approved or standard of care there, even if the tumor itself express telomerase, our antigen. So we are in a way awaiting checkpoint inhibitors or other immune therapy drugs to arrive that in a way show that now the immune system has the chance to do something good for these patients.
I think the immunotherapy, of course, has established itself as a useful tool in clinic through the introduction of checkpoint inhibitors. But I think one thing we sometimes forget, and we're exposed to it very closely because we work with acute myeloid leukemia, for instance, is that the first and probably ultimate immunotherapy has been around already for quite some time. That's called the bone marrow transplant. I mean, we're doing allogenic bone marrow transplants, which basically means you wipe out the immune system, you introduce a new one, but it's never the same. And we know the more distinct it is, the better the reactions are. So if we are able to trigger, say, an immune response, which might be one specific, but sometimes also maybe non-specific, we see that we have results. Why do I introduce that? It's because it will never be only the T cells which will do the job. Otherwise, whoever created us would have just only given us T cells. And we now know that that's not the case. We need natural killer cells, and more emphasis is going on to the natural killer cells. And we also need, for instance, dendritic cells, etc. And I think the multitude of, say, approaches which we currently are developing will probably lead to combinations of these, where we combine T-cell approaches with maybe NK cell approaches or vaccines based on dendritic cell biology, as we are developing. So it's my firm conviction that that's the way forward, and not having the hope for just one approach, which will do all.
Thank you, Jeroen. I think that was a good broad set of questions. So maybe we do a deeper dive and we start with Ultimovacs. Jens, can I ask you a few more specific questions? So it seems that you have a more holistic approach and there's a distinct set of truths and paths. So it's an easier concept to prove, maybe. In this, I mean, Ultimovacs, you do a few things. You locate overexpressed markers and prime the CD4 T cells to target the stem cell. Is this different from conventional treatments that go to other parts of the cancer? And does that help to get around drug resistance?
Well, it depends how you define drug resistance. So very shortly, so we have this UV1 vaccine. It consists of 3 long peptides. So it drives mainly for CD4 expansion, CD4 cell expansion. There's a lot of epitopes within those peptides, so we do not do any HLA screening of patients prior to inclusion in studies. We know that these T cells will migrate through the body and identify their telomerase target presented in HLA class 2 molecules on macrophages, and sometimes also actually HLA class 2 on somatic cells, like melanoma cells. When that happens, they will start to secrete different cytokines that will attract already pre-existing parts of the immune system to the tumor. And since they are re-encountering their target time after time, immunological memory will be developed, and that will lower the hurdle for new immunological reactions, both towards telomerase, but hopefully also towards other antigens. So if a part of the resistance is the microenvironment, we believe that the CD4 cells do contribute to change that in a positive way. And I also would like to say that I totally agree that maybe several parts of the immune system will be relevant in the treatment of cancer. But one important and central cell in that sense will be the CD4 cells, as they are orchestrating much of what happens during an immune response towards infection or cancer.
The UV1 is labeled as a universal cancer vaccine with encouraging survival data in your phase 1 trial in melanoma. Could you please explain what makes it universal and maybe touch on that survival data a little bit?
So universal along different axes. So first, the target telomerase is very common one in cancer. 85 to 90% of all cancer patients indications will express telomerase as a strategy for eternal cell division. So it's a very common antigen. Within the 3 peptides, there are several sequences or epitopes that can be recognized by different HLA molecules. So we do not do any screening of such prior to inclusion in studies. And we have seen that it is a good coverage in different territories globally. So we do not plan for HLA screening in other territories in the future. The third general or global features of telomerase, I would say, is that it is present from very early in cancer development until that late stage, and also in all parts of the tumor. And why is that important? We think that it is important that the antigen persists within the tumor, so that you can build up this immunological memory and lower the threshold for new immunological reactions in the patient. So that is the global aspects of the vaccine. When it comes to survival, we have conducted earlier 3 phase 1 trials in prostate cancer, non-small cell lung cancer, and in malignant melanoma in combination with ipilimumab. Our lead indication is melanoma, and we have in a very small study, though, 12 patients with ipilimumab plus vaccine in metastatic malignant melanoma. We just recently reached median overall survival at the 66 months. Also, we have a 4th phase 1 trial where we combine the vaccine UV1 with pembrolizumab in patients with advanced or metastatic malignant melanoma. We have followed these patients for 2 years, and we see 83% of the patients are alive after 2 years. This is, of course, very early, and that study is with 30 patients. But we have moved into phase 2 in melanoma, fully recruited a trial called Immunicum, where we combine with IPI and Nivo. It's a randomized trial in the US, UK, Belgium, and Norway. And we expect the readout of the primary endpoint the first half of next year in that trial.
Ultimovacs also has a second cancer vaccine candidate derived from the Tet Platform. Could you describe how that vaccine works and maybe how different it is from UV1?
Yes, so that second project or platform is called the Tet Platform. It's in early days still. It's a preclinical project with 1 clinical trial. In this project, we take advantage of a previous infection or previous vaccination. So if you vaccinate a person with the tetanus vaccine, the patient will develop antibodies that recognize a part of the tetanus toxin. We have made a molecule out of those sequences, and to that molecule we can attach different peptides. So the thinking here is that when we inject this into the patients, this will be a more active uptake of the antigens, because already pre-existing antibodies will bind to this molecule and actively take them up in antigen-presenting cells. We have 1 phase 1 trial, first in man or then dose finding study here in prostate cancer to do it as monotherapy. We look for safety, of course, immune responses, and also try to identify the right dose moving forward. And we are currently on the 3rd dose level in that trial. Just want to emphasize that moving forward, you can do this with different peptides. So this molecule that are recognized by the antibodies, to that molecule you combine different antigens, both the telomerase antigens and other cancer antigens in future trials.
And your clinical strategy, it's interesting, right? I mean, you're pursuing ICI and cancer vaccine combinations. How are you selecting the indications and that you want to pursue with UV1? And are you only selecting those that ICIs have been approved so there's less variability?
Yes, so we usually say that we are, of course, following the science, and we are also following the checkpoint inhibitors. It's important for us to develop in indications where checkpoint inhibitors are either standard of care or close to be so, good phase 3 results. And the second thing that needs to be in place is expression of telomerase. In our current 5 indications, the telomerase is used by most, if not all, patients, but there are also some indications where expression of telomerase are lower, for example, in sarcomas. So right now we are developing the drug in different indications where telomerase is present in all patients. Right now, as you know, we are in a way shifting focus in a way. The world has developed the checkpoint inhibitors in the advanced or metastatic setting. Moving forward, I guess much more of the development will be in the early stages of cancer. So, and that is also a very interesting opportunity for us, because telomerase is also expressed in these early stages of different indications.
Thanks, Jens. Jeroen, I'm going to send a few questions to you here. Your lead cancer vaccine, DCP-001, is different from the previous dendritic cells. And I think you kind of touched on this a little bit. Could you go over that more in detail, please?
No, I think it's an important differentiation to make. I think a lot of work has been gone into dendritic cells and how to use them also as an immunotherapy tool. But the way that was done in most cases was to take autologous dendritic cells and then try to educate them, basically. So load them with antigens or expose them to antigens, or have even been approaches to attach cancer cells to the dendritic cell, and these products were given back. But what we've also seen is that often when, for instance, you inject those dendritic cells then back into the patient, intradermally, for instance, the assumption is these would then travel to, say, the lymph nodes, there interact with the T cells, and that's what you get. Then you get your immune response. But that has not been established in most cases. Actually, there's literature enough showing that these dendritic cells, which were, say, introduced back into the patient, just didn't reach the lymph nodes. So how could they interact with the T cells? And this is one of these fundamental things that we also discovered, is that actually what happens is that these dendritic cells, and in this case, the cells we have, they're not traveling. They're just being digested by the host immune system in the skin. Our skin is extremely efficient in mounting an immune response. It's one of our first barriers, of course, so highly trained. And that's what we use. So we're using the principle of, say, indirect priming. So our cells are basically tumor-associated antigens. We made them look like a dendritic cell, and that triggers, say, the immune response, which we need. And we also see the importance of, say, what you call crosstalk and the relationship between the different cells. So that element is key to our principle. And so that's distinctly different from what I would say the classic dendritic cell vaccines. So we try to always steer away from that. Try to explain it again, like, yeah, you cannot really call our product a dendritic cell. It's a cancer cell which we turned into a dendritic cell. Well, we made it look like a dendritic cell for the main purpose to generate a immune response, a very strong immune response. And that does the trick. That's what we see. We see that we generate T cell responses to the antigens which the cell carries. And we see also in our clinical studies that we start to have, say, very promising results on clearing residual disease, showing longer than expected survival for these patients. And that makes it, well, a promising strategy to go forward.
Yeah, that's really helpful. So there've been very many immunotherapies for AML, but you're having some encouraging results with the ADVANCE-2 study. How might the DCP-001 results differ from other immunotherapies?
So we have this indirect priming, so we also need to rely on the host immune system to mount a response. And that's also why the way we use it is not to be direct cytotoxic. And so we haven't, say, developed a CAR T, which is basically a immunological nuclear warhead, which you try to shoot to your target you think is most relevant. No, we allow the immune system to generate a response to antigens which are relevant, which should take control. I think that's one of the key differentiators. That's also why we use it at the time when the patient is initially responding to other treatments like chemotherapy. When then the disease is brought back to a state where it's limited, and in acute myeloid leukemia, you're talking about patients who have entered into a stage which you call complete remission. And that's the time when we start vaccinating. That's the time when the immune system is able again to maybe take control over the residual disease and make it into a durable response. So that's, if you compare it to immunotherapies, a very different approach. But in general, immunotherapy in AML has not been very successful so far. And that has to do with what are you trying to establish? So is there a T-cell response against these antigens? And the answer is probably no. There's too much variety. It's a very heterogeneous disease. There's probably not enough T-cells in the patient themselves to be able to trigger a sufficient response when you need it. That's one. And if you want to go into the route of, say, CAR Ts, then you're challenged with which target are you going to take? If you take a too general target, you basically blast away your white blood cells. And without those, we cannot live, unfortunately. And so it's the challenges so far in immunotherapy to find the best way. And I think maybe the indirect way that we're using could be a very promising way of doing it.
So we also touched on this a little bit too, but the positive results that you're observing in monotherapy studies are encouraging. But is there some potential upside from combination studies, and is that something we're considering?
The answer is yes. And I think what we've seen so far, so we've done a phase 1 study, which was basically in all comers, so patients with acute myeloid leukemia, regardless whether they were in complete remission or refractory to previous treatments. The main observation there was what I just explained, is that you really should vaccinate when there's a state of complete remission. Then you have the best chance to trigger the immune response you need. We now are conducting a phase 2 study, and earlier this year we were able to read out the first responses. And what we are looking at is responses towards the residual disease. So nowadays with new technologies in AML, you can very, very carefully monitor if beyond, say, the blast you can say detect in the bone marrow, whether you can still also detect, say, the mutations which these AML cells carry to a degree which is much more specific than what you could before. So looking at residual disease is key for us to see whether we can see if the efficacy we're looking for is there, and that's what we were able to read out. So to give you an idea, we treated 20 patients with acute myeloid leukemia who were treated with chemotherapy and brought into complete remission, but they were all still suffering from residual disease. Typically, these patients will relapse very quickly. If you look at, say, untreated, the median relapse-free survival ranges from 3 to maximum maybe 6 months. When we subsequently vaccinate them, we saw that in 5 out of these 20, the residual disease completely disappeared. So we had a complete MRD conversion of 5 patients, and on top of that, we had a substantial reduction in residual disease in 2 additional patients. The amount of patients who relapsed were very limited. So we only had 6 out of the 20 who relapsed within the first, say, 8 months. For an aggressive disease like AML, that's extremely promising. But we know that it all depends first what you treat the patient with first. And so we're, of course, looking at combinations, whether, say, the pre-treatment of chemo is the best combination, or whether maybe pre-treatment with alternative regimens might be even more effective. But also in the maintenance setting, it is relevant to see if you can combine it with newly introduced treatments. And one of these is oral azacitidine, and that's something we're looking into as well. Preclinically, we've seen synergistic effects between our approach and some of these approaches. But in clinic, we have to explore that further.
So the survival has been good, but have you been able to measure the duration of DCP-001 induced immune response and the immunological memory?
Well, the survival follow-up in our study is still ongoing, and towards the end of the year, we have a further readout. So we're very eager to see what the data will look like, and eager to report on this, of course. But that's something which I cannot disclose today. But yeah, it has to be seen then how durable it is. Some of these MRD responses I just referred to, they have shown to be durable. And when we then look what the difference is between the patients who have this MRD response versus the ones who don't, then it's almost a black and white. So those patients who convert to become MRD negative, they remain without the disease and alive until we follow up so far. And that's very promising.
We covered so much. Maybe it makes sense if I kind of turn it over to each of you to kind of say some closing remarks.
So we work then with a therapeutic cancer vaccine against telomerase. It's an intradermal injection of the vaccine, totally 8 injections in total. We are currently in phase 2. We have 5 different indications, and in all those indications, we are combining with the checkpoint inhibitors. So we are working in malignant melanoma, ovarian cancer, head and neck cancer, non-small cell lung cancer, and mesothelioma. And for the melanoma and mesothelioma trial, they will have a readout of the primary endpoint first half of next year. So we are crossing our fingers and hope that it will give efficacy to patients.
Jeroen?
The greatness of what we're seeing today, science-wise, is that we learn more and more about the immune system. And it used to be called a black box maybe 10 years ago, and it's still a black box, but there's a little bit more light coming into it, I would say. It's fascinating. So what we discussed earlier, T-cell approaches have made a big step, but it's not solving everything. So I think the whole approach to find other ways of stimulating the immune system is extremely valuable for the patients in the future. So what we are doing, of course, is we're specifically looking into a very hard to treat disease. Acute myeloid leukemia, as just mentioned, is extremely difficult to treat with immunotherapy as well. But it is important because we've seen in AML that the focus has been in trying to find drugs which are killing the cancer blast as soon as you can. But it has turned out to not be sufficient. You still need something after that. You need a durable response. Bone marrow transplant, like referred to, maybe the ultimate immunotherapy, is doing it, but it's still a very challenging procedure. It still carries quite some morbidity. Activating the immune system maybe in a more gentle way, like what we're doing with DCP-001, could be a very good addition to what we're currently developing, and we're very hopeful. So our phase 2 has read out some of the initial responses already, where we have been able to show that you can actually get rid of residual disease using this approach as a standalone, and in combination it might be even more powerful. But we have to see if that leads to increases in relapse-free and overall survival. That data is coming soon towards the end of the year. And so that's a big moment for us. And we hope to be able to report it at one of these key conferences, which is held towards the end of the year. But that is something we hope to disclose soon. But once we pass that hurdle, if we can really show that not only we're able to trigger immune responses, but they're also translating in a longer, say, relapse-free and overall survival of these patients, then we've got something which could be very broadly used. Because the current use in AML maintenance post chemotherapy is one segment. I mean, it's also relevant to, for instance, look at patients who are suffering from residual disease after going through this whole procedure of a bone marrow transplant, because for them there's really nothing left anymore. So yeah, it's very broad, but we first need to pass the first hurdle, and hopefully that comes soon.
Thank you both for taking time. I mean, this is a very complex topic, and there's been a lot of work up to now. I mean, this is like decades of work here. So I appreciate you taking the time, and thank you for our audience for tuning in. Thank you.